Good morning and welcome to one more Greener Talk. This talk is dedicated to our project partner LEITAT. So I would like to welcome Dr. Eduard Borras. Hello, Eduard.
Hello, Jona.
Thank you for accepting our invitation. So I would like you to say a few words about yourself.
Okay, thank you for inviting me for this talk. I am a principal researcher in LEITAT Technological Center in the Department of Circular Economy in the area of environmental technologies, where I lead a group on microbial and electrochemical technologies, where our main research activity is devoted to the development of applied research projects on bio-electrochemical systems. This area of research where microorganisms interact with solid electrodes.
Great, good to have you with us. So we can go directly to the first question. Could you please briefly present the activities that LEITAT is involved in the Greener project?
Okay, so our activities within the Greener project are in several of the work packages, but our main activities, let's say, are on work package 4 on the remediation technologies development for surface water and groundwater technologies. This is a work package that we are leading in LEITAT, and we are very active in participating in 2 tasks for the development of bioelectrochemical systems for treating different pollutants that I will talk later, I suppose, and also the hybrid technologies. And we are collaborating with a lot of partners within Greener in developing several technologies. Also, some of these technologies are then upscaled in work package 6, where we are very active as well. And also, we are active in work package 5 on the soil treatment technologies, supporting some activities on bioelectrochemical systems development. And as well, of course, we are active in the work package 7 on impact evaluation. And I wouldn't like to forget the activities on work package 6 on the ecotoxicology. There is also a group within LEITAT that is working on the toxicology assessment of the technologies that we are developing within the whole project.
Thank you, Eduard. So if we can go to the next slide. As LEITAT is leading the development of bioelectrochemical systems and hybrid technologies for contaminated water, could you please present the advancements of your work there?
Yes, of course. So let's start with abiotic metal removal. This is one of the technologies that we have developed in LEITAT. So we constructed a specific bioelectrochemical reactor where we remove metals abiotically, taking advantage of the reactions taking place around the cathode. And we support this process with a biofilm grown on the anode to reduce the overall energy demand. We have applied this technology under different running electrochemical modes for different target metals. We have studied it, the removal of these metals from synthetic waters perspectives to study the processes of the metal removal, but also using metallurgical industry polluted groundwater supplied by our partner Tau. You can see here that in this slide some picture of the experiments where we recover copper in the surface of the electrode, but also as precipitates within the buffer tank that we have in the system. Also, I would like to mention the activities from our partner, the University of Surrey. They are working on a different approach, promoting the biological removal of metals in a biocathode with different approaches. And we are collaborating with them to benchmark the biotic and abiotic metal removal processes. And as regards to the hybrid systems, so the activities that we are doing in LEITAT on metals removal via abiotic processes, this technology will be coupled with the wetlands that are developed from the project coordinator of the University of Burgos. In Burgos, they are developing wetlands for the removal of metals. And we want to couple these 2 technologies using the bioelectrochemical reactors as a pretreatment for softening the metals content before entering the groundwater polluted from metallurgical sites inside the wetlands. Another technology that we have studied within Greener project is the removal of antibiotics using a BES reactor. In this case, we have focused on the acclimatization of an electroactive biofilm grown on the anode for the removal of 3 target antibiotics. We have acclimatized this biofilm to very high concentrations of the 3 antibiotics, and we have assessed the microbial population evolution over time. Also, we are studying the presence of antibiotic-resistant genes within this biofilm, and we are studying the metabolites that are being produced and the changes in toxicity when we are applying the BES reactor compared to conventional biological biofilm processes for removal of these antibiotics. Another approach for the antibiotics removal that we have studied in LEITAT is a hybrid system. It consists of a constructed weather that incorporates bio-electrochemical systems inside the gravel matrix. For these experimental setups, we have used 2 approaches, a tubular approach and a granular approach. We have tested these 2 systems also for the removal of the 3 mentioned antibiotics under different operating conditions, but at a very low concentration, more similar to the concentrations that we can find in a wastewater. Another technology that we have developed in LEITAT has focused on the hydrocarbons removal, stimulating anodic-related biodegradation processes. For this, we have been collaborating with Tau, our partner in Greener, and they provided us some groundwaters polluted with different types of hydrocarbons from an industrial site here in Spain. And from those samples, we could isolate in our lab-scale experiments microbial communities capable of removing hydrocarbons and also being electroactive. And with the data gathered at lab scale, we are now constructing a pilot that will be soon installed in an industrial facility and will allow us to demonstrate the feasibility of this technology for removing hydrocarbons on site. Also, another technology that we have been developing within Greener deals with the lindane removal. For this, we have developed together with Tau a technology that removes lindane abiotically in a reductive process related to a cathode. And this cathode is supported by a bioanode that is supporting part of the energy that is necessary for the removal of lindane from the removal of organic matter. Also, we have been collaborating with the University of Cagliari, our partner in Greener, for the modeling of different processes. Here you can see some images of the first activity that we developed together for the modeling of the anode-associated processes. And this has served as a starting point for further modeling of different remediation technologies and developed within Greener.
Thank you, Eduard. Great collaborative work was performed here. And with this being said, I will move to the next question regarding the pilot scale experiments for water technologies and the ecotoxicological analysis of the upscale technologies. So could you please elaborate more on any challenges and achievements reached so far?
Yeah, well, the evaluation of the technology's performance that we are having in Greener is not only focused on the target pollutants removal. So we want to go further. So it's not only about reaching the concentration levels that the regulations are marking for each water or soil conditions, but we also want to focus on the technology evaluation of the different bioremediation technologies. So even at lab scale, in Work Package 4, Work Package 5, we are studying how the toxicity is changing when we are applying the different studied technologies. And this is an activity that has been jointly developed from our coordinator at the University of Burgos together with LEITAT. where we have implemented different techniques in vitro and in vivo, either for soil and water, where these technologies or the different technologies have been evaluated in terms of ecotoxicology changes before and after the treatment with the technologies.
So I know that there were some unexpected findings and there is room for further research activities, right? So if you can elaborate more on these.
So during the GREENER project, so unexpected opportunities of collaboration have appeared. It has been the case with the Institute of Technology of Carlow, our partners in Ireland. They are developing eco-piles for the removal of petroleum hydrocarbons from polluted soils. And they have a pilot installation in an industrial site. And we had the opportunities to incorporate graphite roads inside the eco-pile. And now we are evaluating the treatment efficiency when we are incorporating graphite roads inside an eco-pile for the removal of petroleum hydrocarbons. This wasn't initially expected within the project, but it's always good to take these opportunities of collaborating. Also, during GREENER project, we could study an important issue when someone wants to upscale the bio-electrochemical reactors in a modular approach. So using different reactors connected in series. And for that, we developed systems to control the voltage balance within these serially stacked bio-electrochemical reactors that was based on the use of diodes. With this control system, we could specifically and very accurately control the electrodes potential in the cells over time and controlling the excess current through the diode. And this system allowed us for the further modular upscaling of any kind of these bioelectrochemical systems reactors that need to be connected serially.
So thank you again for your time and for your presentation. I'm looking forward to more results to come until the end of the GREENER project. So see you all at the next GREENER talk.
Okay, Ivana, thank you very much for giving me the opportunity to present the activities from LEITAT within GREENER project. And I'm really willing to have the opportunity to present you the next advances before the end of the project.
Thank you. See you all at the next GREENER talk.
See you.